Welcome to Sport Fishing Television. I'm Chuck Myers. On today's show, we're heading south to Carlsbad, California to do a little research. We've been invited to take a video tour of one of the most interesting facilities in Southern California, Hub's SeaWorld Research Institute, with our co-host, senior biologist Mark Drawbridge. Hub's research focuses on gaining scientific knowledge and finding practical solutions to one of the most critical conservation challenges facing marine ecosystems and species. As you're about to see, right here on Sport Fishing Television. We'll be right back. Sport Fishing Television is sponsored by Yo's Custom Rods, come and experience Alaska at Anderson Island Lodge. AllCoastSportFishing.com, real-time reports.
The smallest moments can have the biggest impact on a child's life. It takes a man to be a dad.
Welcome back and thanks for joining us. This show's about conservation and the white sea bass. Hub SeaWorld Institute was founded in 1963 by Dr. Carl L. and Laura C. Hubbs. Hub SeaWorld Institute program was created to investigate ways to counteract the depletion of California coastal marine fisheries through stock enhancement. Hubs has a unique partnership between California state resource agencies, public utility companies, volunteer user groups, and the scientific community. One of the few stocking programs that assess the biological and economic impacts of its releases. What you're about to see is the only program of its kind on the west coast of North America, saving the white sea bass.
My name's Mark Drawbridge. I'm a senior research biologist for Hub SeaWorld Research Institute, and we're here at the Leon Raymond Hubbard Junior Marine Fish Hatchery in Carlsbad. On site here we have a marine fish hatchery where we raise hundreds of thousands of white sea bass for release back into the ocean. So we'll be taking a look today at that process. So this program, like I said, has been around since the early 80s. This particular facility was constructed in 1995, so we've been here for just over 10 years now. And in terms of enhancement programs, there's quite a variety and large-scale enhancement programs, but they're primarily geared towards freshwater fish and salmon. As far as marine enhancement programs go, this is one of the largest in the nation. And an interesting one as well in terms of just the support of the community, in particular the recreational fishing community. And I say that because the funding for the program comes from the sale of sport and commercial fishing licenses, and the majority of that is from the recreational fishing community just because of their sheer numbers. So in that regard, the fishermen are footing the bill for the program, and they're also actively involved in the program in the sense that here at the hatchery we raise the fish to about 3 or 4 inches and then tag the fish and then deliver them to different grow-out facilities located up and down the coast. And the volunteer fishing groups take it from there, and they'll raise the fish from 3 or 4 inches to 8 to 12 inches before they're released. And then once they're released, the fishermen have a real strong incentive to help us get information back on those fish. As I mentioned, every fish is tagged in the cheek muscle, so we have a pretty extensive program coast-wide to try and get the heads of the fish back so we can evaluate how well the program's working.
How is the program working as far as the returns that you're getting?
The program's working very well. Over the years, we've gotten back just over 1,400 tags. The majority of those have been sub-legal sized fish, so we have researchers that go out and survey the short fish. But every year we're seeing more and more adult fish come back. And that reflects kind of the increase in the scale of our releases from what used to be tens of thousands of fish per year. We're getting up now into the hundreds of thousands of fish per year, and we're starting to see that reflected in the catches. So just last week, for example, we got a fish back that was 12 years old. So it was actually spawned in about 1994.
Wow, and it was from here?
And it was from here, sure. Actually, it predates this facility, so it's pretty exciting. Every tagged fish that we get back tells a story.
Mm-hmm. And talk about that a little bit, about the story itself. What do you really get out of this information when they come back to you?
Well, the tags have a code etched into it, so we can plug that back into our database, and it'll tell us exactly what group of fish that came from. So then we can start to understand how far that fish has traveled, and really what we're trying to get at is look at the survival rate over time and to see how we can optimize that in terms of how big the fish need to be, where's the best place to release them, what time of year is the best to release them, those types of questions. So it's really a very large-scale research program.
Now, you told me earlier that Hubs is getting more involved with other species, not just white sea bass. Let's talk a little bit about that.
Right. Well, our research is focused in a couple different areas. What we're interested in doing is looking at how aquaculture, which is basically fish farming, can be used as a conservation tool. And there's a couple approaches that we can use. With the white sea bass program, we're raising fish and then releasing them back into the ocean to restore those depleted stocks. Well, those very same depleted stocks, the reason they're depleted is because everybody likes to catch them and eat them. So another way that we can help is in some cases, raise those fish directly for market if somebody wanted to do it commercially like they do with other species like salmon. And by doing that, the consumer has that product readily available and it takes pressure off the wild fishery. So in that regard, we're looking at other high value species like California halibut, California yellowtail, California sheephead we have on site here. So those are some of the examples of the fish that we're looking at.
What about cowcod?
Cowcod's another one that we actually started a program up about 3 years ago that's focused on some of the deeper water rockfish that are in a lot of trouble. Cowcod is one of those and Boccaccio is another. So we've had some success over the last couple years figuring out how to catch those from several hundred feet of water and bring them up and have them survive in good conditions. So at our other laboratory down on Mission Bay, we actually have captive breeding populations of Boccaccio, cowcod, and vermilion rockfish.
And how are they doing as far as survival and research, as far as what you're doing with white sea bass? How's that program working out?
It's working out real well. It's at the early stages of development, and so with each new species, you've got to work out details along the way. So the first step is to get the broodstock into your facility. These are the breeding fish. Get them acclimated in such a way that hopefully they'll reproduce in captivity. Those particular fish have not yet reproduced for us, but they've only been there for a year or 2 years. So we're optimistic that they will mate in captivity, but only time will tell.
So how many pens do we have out there?
Well, we have 14 different facilities. This is an example of one right out here that we happen to own and operate. We put this out there a couple years ago, and so just as an example, it's a very simple design, just a floating net pen. You know, the fish go in the central containment net, and then there's a predator net to keep things like seals and sea lions out on the outside. This is a particularly nice setup out here because this is a private property that's owned by the power company, Cabrillo Power, so they keep it secure for us. And the property as well that the hatchery is on is owned by Cabrillo Power, and we're very fortunate that they lease it to us for $1 a year, so it's one of the other things that really makes this program work. This is an example. Like I said, there's all different shapes and sizes. This is one of the larger ones here. It's actually a 2-pen system, and but it's pretty typical. It's based on same technology they use for the salmon industry.
So the ones that are in the pens, they come up from your hatching areas out upstairs here.
Right.
And then you put them in there until they get to a certain age.
Right. So what we do is the hatchery facility here, we pump water and we're raising the more sensitive life stages. And to do that, you need really high quality water in order to produce the eggs and the larvae. As they get bigger, their requirements for that high quality water diminish somewhat, so you can start to look at acclimating them to natural conditions. So we'll raise them in the hatchery till they're about 3 or 4 inches, and in this particular case we just float a pipe along the water and sluice the fish out in the pipe right out into the pen, so they always stay in the water. Wow. It works out pretty well. In other cases, facilities that are further away, we have to load them into a truck and drive them up the freeway and then sluice them out of the truck. Or in some cases, like Catalina, we have a couple facilities, we'll have to drive them up the coast, load them onto a boat, and the boat takes them over. So it's a pretty intensive process, a little bit stressful for the fish, but over the years we've just worked it out and really have it pretty well dialed in right now.
Right on.
Yeah.
Okay, let's go take a look at the facility.
So this is just a map of the coastline here to show you the locations of the different grow-out facilities that we have. So we're right here in Carlsbad at the hatchery, and this is the pens that you saw outside. And so you can just see as you move up the coast the locations of the different facilities. So this is really the range of the program that we work in, and that's kind of driven by the funding, which comes from Southern California. But as far as the fish is concerned, one of the challenges that we have working with white sea bass is that these fish, they don't recognize any boundaries, and once we release them, they just take off. So you figure out pretty quickly how big the Pacific Ocean is. But nevertheless, we've been getting some good results with our tag recoveries back, and kind of the story's starting to unfold, so it's pretty exciting. Anyway, just a lot of infrastructure here to support the operations. A lot of plumbing. As you can see, we keep the PVC guys in business. This particular system here has been pretty valuable to us. We constructed this about 3 or 4 years ago. It's an ozone system. It ozonates all the water, which disinfects it before it goes to the eggs and larvae in particular, which are very, very sensitive to any kind of contaminants that might come in the water. So that's really helped our survival rate, just as an example. So the egg collection process for sea bass, and actually many of these species that we work with, is really pretty straightforward because they're pelagic spawners, which means they just release their eggs into the water column. So what we do is we just, as the water is flowing out of the tank naturally as part of the recirculation system, we pass it through a mesh bag. And so the eggs would be retained in the bag, the rest of the water gets pumped through. So each morning, the fish usually spawn in the early evening. The following morning, we just come through and look to see if there was a spawn that evening. And then we decide if we're going to set that up for production or not. So that's the process, and it's the same process we use for California halibut, California yellowtail, and California sheephead. Some species like cabezon and lingcod will lay egg masses, which require a little bit of a different technique, but it's still, the mating is the hard part. Once you get them comfortable in captivity and they're mating for you, then you're off to the races. So that's the biggest challenge. Our hatch rates are usually up over 70%, so 70% of the eggs will hatch. And then in the early stages, you incur pretty heavy mortalities, even in a culture system. But at the end of the day, you're still left with tens of thousands of larval fish that turn into juveniles. So you're still working with pretty good numbers. Now each fish that's in here, we have a little pit, what's called a pit tag, in their dorsal muscle, and when we scan that fish, we get a 9-digit readout. So each fish has its own, basically a social security number that we can track that fish for the entire time period that it's here with us. And in that regard, we know where that fish was caught, when it was caught, when it was last weighed and measured. And also we're working simultaneously with some geneticists, and we're able to, using genetic techniques, identify who the parents were by sending samples of offspring to the genetics lab. So in effect, each parent that goes in here has a genetic fingerprint associated with it. So that's some of the other technology that we're taking advantage of. Yeah, we'll give them sardines or anchovies. We do give them squid. It depends on the day. This particular day, they're getting fish, and I think they're injected with vitamins. So we'll add supplement vitamins just to make sure they're getting everything they need.
Okay, you want to show us some more stuff?
Sure. So this just shows you kind of graphically a few different things. This is a beaker that has eggs in it. So the eggs, the good eggs, what we do is the good eggs float, and then there'll be a percentage of eggs that sink out. And so this is the stuff that we're after here, is the good floating eggs. And so you can see here's a picture of an egg here with the embryo forming inside. So it's a pretty neat process. You know, we're able to watch it here in the laboratory. It's pretty spectacular. This shows you the spawning profiles for the 4 tanks that we have here. And it's a little bit hard to see, but these red spikes represent spawning events, depending on the water temperature and the day length. And if you were to overlay all of these together, time here is on the y-axis, you'd basically see that we have eggs being produced all the time. So it keeps us really busy.
Okay.
Just from a research standpoint, you know, it's really interesting because most of us are biologists, but a big part of this is also you get into the engineering of different types of systems. So as you look around, you can see all different types of filters and pump systems. And this is, again, another part of our research where we're interested in optimizing these systems for the fish in particular, but also just looking at things like energy efficiency and those types of things. So again, it's beyond the biology, it gets into the engineering aspects. So we're collaborating with other researchers around the country who specialize in these areas.
What do we have going on right here?
Okay, so this in particular is a type of biofilter, and it's called a biofilter because the filtration that's done is done by bacteria, so a living organism. And this is a standard part of any type of recirculation system, although there's different ways of doing it. So what you have here is a moving bed of media that bacteria, naturally occurring bacteria, adhere to. And those bacteria in a recirculating system convert the waste product of the fish, which is ammonia, into a non-toxic form. So the problem when you have a lot of fish in a small volume of water, if you let that waste product build up, it would become toxic to the fish. So this is one way of naturally detoxifying that. So it works out very well. So basically what we have here is some of the juvenile fish. These fish are about 40 days old or so, and these are the smallest fish we have on site right now. There's probably, I'd estimate, about 10,000 fish in here. So this would be one pool. Right now we have 3 pools from this particular crop of fish holding white sea bass. So nothing really fancy going on here. You know, we're just providing the fish with some high quality water, and we've switched them on to a high quality feed. So they'll spend another few weeks in here, and then we'll move them to a different system, and then they just kind of move through the process as they get bigger and bigger. So ultimately going into the net pens that you saw, and then they'll be released into the wild. So these fish have a ways to go, but this is what a baby white sea bass looks like. One of the biggest challenges we have during this stage is actually cannibalism, where if you look in the pool, you can see a select few fish that might be twice as big as their siblings. And the reason they're twice as big is because at some point during the culture process, they've been successful at eating their siblings, so they just get bigger faster. So we have a pretty rigorous grading program where we periodically pass the fish through grader bars and sort them by size, so we can't get that disparity. So for the most part, these fish that you're looking at are very uniform in size.
So when they get a little bit bigger, you know they're eating their brothers and sisters. You pull them out immediately to another area? Is that how it works?
Yes, we do. We isolate them to the extent that we can. But that just kind of shows you that these fish, they're fish eaters. That's what they're programmed to do. So in that regard, it's a very natural process. The densities that we run here are much higher than they would be experienced in the wild. Although cannibalism is also reported fairly commonly in the wild as well. So the sex of the fish, you can tell when the fish is about 6 to 8 inches in length. And one of our actually our pathologist has been looking at that, because sea bass are pretty interesting in that they're in the croaker family, which means that they actually use their swim bladder to make a croaking sound. So as part of that process, that's specific to the male fish, and so the males have a specialized muscle that if you were to dissect the fish open, you could actually see that muscle, and that's how we can tell fairly early on whether it's a male or female. Okay, so this is what we call our outdoor raceway area, and it's just an extension of the hatchery facility. It's kind of an intermediate between the fairly sophisticated recirculation systems indoors and then the cage systems that they'll be going into after this. So these are really simple design systems. The water comes in at one end, it's pumped from the lagoon, passes through some sand filters, and then it goes out the tail end of the raceway. So this is really the final staging area for the fish here. They'll be tagged out here and then loaded from these systems, usually put into the truck and then delivered to wherever they're going. So
how old are these fish in here?
These particular fish are probably about 8 or 9 months old actually right now. So these fish are large enough actually to release. So we're kind of in a they'll be leaving here very shortly. So really all we've done is, as the fish have gotten bigger, we've moved them into larger and larger systems, just giving them more water volume, basically.
At this point, what are they feeding on?
They're feeding on an artificial diet right now. They're still on a pelleted diet, which they'll be on right up to the point of release, actually. It's just kind of a standard fish feed in a pelleted form. High quality, so that they do real well on it.
Now, are these guys sick, or are they just...
Yeah, the tendency will be if the ones that don't spook away from you are the sicker ones. So they'll be culled out. Well, densities in here are pretty high, so you can have different aggressive interactions, and but you're looking at a handful of fish out of probably 10,000. So the reaction that we want to see from them is what the majority of the population has done, and that is move away. Yeah. So it's a good sign, and when we release them, in fact, working with the net pens is really nice because, and it's really unique to this program, because a lot of programs will take them straight from the hatchery, and they're working with a much smaller size too, and they'll just put them in a truck and take them to a release site and dump them into the water. Well, what we found is that if you can put them in a cage and kind of acclimate them to their natural conditions, and then just lower the net down and let the fish swim out, it works very, very well. So it's really a nice addition to the program.
Mark, one thing that we forgot to ask you is, that white sea bass have this stone in their heads. Can you talk a little bit about that? How does that happen? What does it use? What do they use it for?
Well, yeah, you're talking about the otoliths, and that's just part of the inner ear of the sea bass and other fish. And the reason those things are so important is because that's how you age fish. That's the best way, the most accurate way to get an age off a fish. They each year as they grow, just like a tree, it leaves a ring. And so you can section those in half and then look at them under a microscope and count them and see how old the fish is. So for us, it's really valuable and important to get those when we get the heads of the fish. And I say that because then we can look at, especially for the tagged ones, we can kind of validate what we think the age of the fish is with what we know the age of the fish is based on the tag that's in the fish. So I know some folks are reluctant to give those up because they make really nice jewelry. We do have a capability to do an exchange program. So we can give you another set of otoliths that will almost identically replace those in the head of your fish if that's something that you want to do. So again, if you can leave the stones in there and if you really want them, just let us know and we'll give you a replacement set.
Let's talk a little bit about how the fishermen can get more involved in the program here.
Well, the fishermen, for us, the biggest help the fishermen can be at this point in the game is really helping us assess how well this program's working. And to do that, what we need the heads of these fish back. So what we've done is we've set up collection points at most, if not all, of the landings up and down the coast. There's freezers that are set up at those locations that should be labeled for receiving the white sea bass heads. So any fisherman that catches a white sea bass, they can take the good stuff home, take the fillets home, but if they could save the heads for us, drop them off at the landings, we'll scan those fish and get all the information we need from those fish.
Do you have a website that people can go to?
Yes, we do, actually. Our institute website is www.hswri.org. Lists all of those drop-off locations, so it's a good source of information. We have a hotline to get information on where you can drop off white sea bass heads, and that's 1-877-SAVE-WSB. We do offer tours, so we do have a docent program where folks are allowed to tour through the facility. It just needs to be scheduled in advance, but we certainly would offer that.
The first experimental release of more than 2,000 juvenile white sea bass took place in October of 1986 in Mission Bay, San Diego, California. Since then, the program has released more than 300,000 juvenile white sea bass into embayments and near-shore coastal areas of Southern California. Well, that's all the time we have left. We hope that you've enjoyed watching the show. Be sure to visit our website at www.sportfishingontv.com for our shows and podcast downloads. Please help us get the word out. Tell your fishing friends about us. We'd like to thank Mark Drawbridge and Karen Terra for making our tour possible. It's places like Hubs that is making a difference in conservation. When you can, do your part. Practice CPR, catch, photograph, and release. For our children and their children. I'm Chuck Myers. We'll see you next time on Sport Fishing Television.